Good afternoon, everyone. I am Dr. Erica Ullman Sapphire, Professor, President, and CEO of the La Jolla Institute for Immunology, or LJI, as we like to call it. It is a great pleasure to welcome you to this special edition of Live from the Lab. Today's event is made even more meaningful by the generous support of the Global Autoimmune Institute, abbreviated GAI. We have already received an overwhelming number of questions and interest in this particular webinar, and I am grateful to each one of you for joining us today. Thank you for carving the time out of your busy schedules to learn, engage, and be part of this conversation on this extremely important topic. GAI is an organization that is redefining what it means to drive and fund research in autoimmune disease. They don't just invest in research, they invest in everything that transforms research into real-world impact. The leadership that leads to discovery, the infrastructure that accelerates progress, and the platforms that connect scientists, clinicians, and patients to turn breakthroughs into treatments. Their strategic, holistic approach to philanthropy is accelerating progress in the field, and today's program is a perfect example of that. GAI is a powerhouse in the autoimmune disease space, not only advancing research, but also bridging the gap between science and the patient community. They are driving scientific literacy, empowering individuals to navigate their own care with knowledge and confidence, and connecting patients with groundbreaking discoveries that are actively reshaping the future of autoimmune disease treatment. We are especially honored that GAI's Executive Director, Sandy Boek Werness, is joining us today, not just as a guest, but as our moderator for today's Q&A and program close. Sandy is a visionary leader in healthcare policy and advocacy with a career that has been dedicated to making thoughtful, lasting change in how autoimmune diseases are understood and treated. She brings a rare and powerful combination of expertise in public policy, legal strategy, medical research, and nonprofit leadership, and her impact on this space cannot be overstated. She is a longtime advocate for patients and families affected by autoimmune disease. As a driving force behind critical research initiatives, and she is a respected thought leader in the philanthropic space. As the founder and executive director of GAI, she has worked to ensure that scientific research translates into real-world solutions, connecting cutting-edge research with the realities of patient care. And now, as a board member of La Jolla Institute, Sandy is deeply involved in the advancement of immunology research. She's helping to shape the future of LJI, not just as a funder, but as a thought partner in how we connect scientific discovery to real-world impact to patients. It is an absolute pleasure to have Sandy here today to help lead the conversation, and we're so grateful for her guidance and support in making today's program possible. Before we dive into the presentation, I want to take a moment to introduce LJI for those of you who may be joining us for the first time as part of GAI's network. So let me share my slides with you. And let me push play. So LJI is a very special place. It is a nonprofit private research institute. We are entirely focused on human immunology and the human immune system. We exist for research. We are driven by scientists for scientists. We have this focus on the immune system because nearly every human disease is controlled and sometimes caused by that human immune system. Study of our own immunology affords the greatest opportunity to positively affect human health. We are small, but we are mighty, and we are ranked top 5 in the world in this space. We have lots of breakthroughs in progress. You know, the immune system touches nearly every disease. We are making incredible advancements in precision immunotherapy against cancer, the cure from within. We are understanding the role of your immune system in cognitive health, in Alzheimer's, Parkinson's, other diseases that keep our brains healthy as we age. We are leaders in understanding defenses against microbial threats, understanding how our DNA changes as we age, and uncovering new targets and opportunities in autoimmune disease. And we have 7 assets in clinical trials right now. We are 420 total staff, including 200 award-winning PhDs and MDs. We hire the best in the world, and they come to us from over 35 countries. And something that I'm very proud of is the exceptional culture and collaboration that exists within this building. We have been ranked best workplace in San Diego by anonymous employee surveys every year since 2016, and we're really proud of that. Our researchers are organized into 4 centers of focus: autoimmunity and inflammation, cancer immunotherapy, differences between men and women and our immune systems that help us better discover the causes and mechanisms of disease and treat them better as well, and also our Center for Vaccine and Immune Innovation. We pursue new treatments, A to Z, everything from aging to Zika virus, all of the diseases that you see here, and so much more. It really is an exceptionally innovative place at the forefront of discovery, and my own research has transformed after moving here 6 years ago. So I'm very pleased to welcome you to today's webinar and to our institute and our partnership with GAI. With that, it is my absolute privilege to introduce today's featured speaker, Dr. Sonia Sharma. Sonia is a scientist of extraordinary vision and impact. She's an associate professor at La Jolla Institute for Immunology, where she serves as the director of our Functional Genomics Center and director of our center to uncover the sex-based differences in immunology, the differences between men and women in our immune systems. She is a Harvard-trained immunologist. She also began her academic journey at McGill University before Harvard. McGill is one of Canada's most premier prestigious research institutions. And at that institution, she laid the foundation for what has become a career of groundbreaking discovery at the intersection of genetics, immunology, and disease. Dr. Sharma's research is powered by an insatiable drive to decode the immune system's most elusive mechanisms. She is trained at the cutting edge of genomics science. She uses high-throughput technologies like CRISPR, RNA interference, and metabolomics to dissect the earliest immune signals that shape disease outcomes. Her work is redefining our understanding of autoimmune disorders, not just in terms of which cells are involved, but when and how the earliest whispers of disease take root. And in challenging the long-standing assumption that immune cells alone orchestrate autoimmunity, she has uncovered a surprising truth. Non-immune cells, such as skin cells, vascular cells, and epithelial cells, like skin cells that line every tissue and vessel, they act as the body's first responders, dictating the immune system's course long before symptoms appear. That fundamental shift in perspective has profound implications. By identifying those early immune triggers, Dr. Sharma's work could allow clinicians to predict who will develop autoimmune disorders before the first symptoms arise. It also opens the door to a new era of precision medicine, one that moves beyond the broad immunosuppression that's been traditionally used for autoimmune disorders, and instead toward what we want, which are therapies that are more targeted and more effective with fewer side effects. This is really important. As director of LJI's Center for Sex-Based Differences in the Immune System, Dr. Sharma is also tackling one of the most overlooked questions in immunology. Why do men and women experience disease so differently? Women are much more likely to develop autoimmune disorders, while men are more susceptible to infectious diseases. Yet the molecular underpinnings of those disparities remain very poorly understood. But there's a lot of insight that we can use and harness there. Dr. Sharma's research is illuminating those differences at a biochemical level. She has identified bioactive lipids, powerful signaling molecules that show up in dramatically different numbers in men and women with Alzheimer's and with inflammatory diseases. So both diseases with immune system involvement. And that finding is a lot more than academic curiosity. That finding is a major step toward unlocking sex-specific treatments for a range of conditions, from autoimmunity to neurodegeneration. Her expertise in human genetics, immunology, and metabolism has made her an indispensable voice in the field. She serves on study sections for the National Cancer Institute, the National Institutes of Health, and leading scientific journals, and that's an honor reserved for the most trusted minds in biomedical research. During today's presentation, Dr. Sharma will cover a range of topics with three main foci: the fundamental mechanisms behind autoimmune diseases and why early immune signaling matters. 2, how genetic and metabolic factors influence autoimmune disease and risk and progression. And 3, how current treatments suppress the immune system broadly, and what her lab is doing to develop more targeted, patient-centered alternatives. Following her talk, we will open the floor for a Q&A session moderated by Sandy. We encourage you to submit your questions throughout the presentation via the Q&A function in Zoom, and we will do our best to address as many as possible. And with that, it is my pleasure to introduce Dr. Sonia Sharma.
Thanks so much, Erica, for that really kind introduction. And thank you to the GAI for bringing this live from the lab presentation to you all today. With that, I'm going to share my screen. So as Erica mentioned today, what I'll be speaking to you today is entitled The Autoimmune Primer. And that is decoding early immune signals and their significance to disease, and in this case, to autoimmune diseases. So let's get right into it. March is, of course, Autoimmune Disease Awareness Month. And with that, I thought I would start by introducing autoimmune diseases. An autoimmune disease is classically defined as an inflammatory condition in which the body's own immune system inappropriately attacks healthy organs or tissues. Some more common examples of autoimmune diseases that you may know of are lupus, rheumatoid arthritis, psoriasis, Hashimoto's disease, and others. But they actually comprise more than 100 individual distinct autoimmune diseases. You should know that each individual disease is often considered relatively rare by itself. However, collectively, autoimmune diseases are not rare. They actually collectively affect, excuse me, we need a presentation mode. They actually collectively affect at least 20% of Americans. And this translates to about 4% of the world's population. These diseases, you should know, are also significantly increasing in incidence. And part of that might be because clinicians are now better at diagnosing these diseases, but part of that also appears to be that they are in fact increasing, really emphasizing the need for new and better treatments for these diseases. So this slide is indicating the affected organs by different autoimmune diseases. And you should know that autoimmune diseases pretty much affect almost every organ system in the human body. And some of them, like lupus, for example, can affect many organs at once in the same patient or in the same family, indicating that there's a strong genetic component to autoimmune diseases. This often makes them difficult to treat because in affecting different organ systems, this just makes treatment just that much more complex, especially if a clinician has to address more than one disease or disorder in a single patient. And as a result, as Erica mentioned in the introduction, many of the traditional autoimmune disease treatments rely on very strong immunosuppressants that basically just serve to completely eliminate or completely suppress the immune system in order just to alleviate that inflammation. This is a good thing for alleviation of inflammation, but these powerful drugs can also render the patient immunosuppressed and therefore very susceptible to infections by bacteria or viruses. And that's not a good thing long term. A lot of these powerful immunosuppressants also have toxic side effects, and patients don't like to be on them chronically. So it's really like playing whack-a-mole, and it's really like waiting until the damage happens and the autoimmune flare-up occurs, and then going in, completely suppressing the immune system, waiting for that. Can we go off these drugs? Is the patient okay? So not really a tailored treatment, but really a one-size-fits-all treatment that doesn't really fit for everybody. And thus, we believe that as immunologists, that a better understanding of autoimmune disease is necessary to develop better, effective, and more specific treatments for these diseases. So what are the risk factors for autoimmune diseases? Well, we know after years of observational or epidemiological studies of patients in the US and globally, that autoimmune disease arises as a combination of genetics, environment, and of course, immune system dysregulation. Regarding the immune response, a normal immune response is solely focused on attacking and invading pathogen, a bacteria, a virus, or even a tumor cell. By developing immune arms, or for example, antibodies. Antibodies are a response of the immune system that specifically seek and destroy the pathogen, whether that's a bacteria or a virus. Under normal conditions, this is limited to the pathogen. It's limited. The antibodies don't recognize healthy tissues or healthy cells. But in the case of autoimmune disease, something goes wrong. The immune system mistakenly produces autoantibodies, for example, that search out healthy tissues instead of pathogens. And this is where the immune system attack occurs and results in inflammation and tissue damage. That's one example of immune system dysregulation in autoimmune disease in the form of autoantibodies. But the immune system is actually very complex, as anyone at LJI will tell you, endlessly if you like. And autoantibodies are just one piece of the immune system puzzle. The immune system is actually divided into two distinct arms, which we call the innate immune system, or the early immune system, shown on the left of this slide, and the adaptive immune system, or the later immune response, shown on the right-hand side in green. Autoantibodies and autoreactive T cells are part of the later or adaptive immune system. But if we look on the left and consider the innate immune system, or the early immune response that occurs before the appearance of autoantibodies, for example, if you look at the bottom of the slide where I've shown you the several different innate immune cell populations, you can immediately appreciate that innate immune cells actually outnumber adaptive immune cells. And if you think about innate immune cells, for example, like neutrophils, which are part of the earliest immune response to a pathogen, these are actually the most abundant immune cell in your blood. And they're very important in representing the first line of defense to protect us against virus and bacteria. As Erica mentioned in her introduction, many important studies of autoimmune disease have focused on the production of autoantibodies and adaptive immunity, because of course, these are the end stage of the disease process. But my lab and others believe that innate immunity, because it's the foundation of the later adaptive immune response, is equally important in the autoimmune process. This idea was basically a concept until the last 20 years of genome research on the human genome. Remember that the human genome was fully sequenced in 2003. And after this was done, it empowered researchers to obtain more and more clues from human genetics. Because once the genome was sequenced, we actually knew what we were dealing with, how many genes we were dealing with, what type of genes, what is their sequence. So we could track these genes and associate them with different diseases. And one of the, I think, surprises about human genetics research over the last 20 years with respect to autoimmune disease is that innate immune genes appear to be strongly associated with the development of autoimmunity. So in this slide, I'm showing you different boxes where each box represents a different autoimmune disease, lupus, RA, and others. And in the italics, the italicized terms represent a gene, an innate immune gene expressed in neutrophils and other innate immune cells that is associated, where mutations in this specific gene have been shown to result in autoimmune disease in humans. And the bolded italicized genes are shared between different autoimmune conditions. What does this tell us? This is real human genetics data telling us that innate immune activation, the early immune response, is common and important to many different autoimmune diseases. My lab actually specializes in studying innate immunity, and we do this through the lens of metabolism or metabolites. Shown here is a representation of what we call the central dogma of biology, in which genes, in the form of DNA, produce RNA, which result in proteins. And proteins do all the work in the cell. But all that work, what is the point of it? Well, the point of cellular biology, cellular function, is really cellular metabolism. Our cells and our tissues need energy to grow, but also to perform all of the essential functions that they do within our body. And so proteins really function to regulate or produce metabolites. Metabolites are the small molecules that regulate energy consumption and energy usage in the cell, including in immune cells, which are very highly metabolic. My lab and others are finding through the study of metabolism and metabolites that metabolites, being at the end product of the central dogma of biology, are very intimately linked, that changes in metabolism are very intimately linked to human disease processes. Not only that, but because metabolites are small molecules, they're relatively easy to manipulate in terms of their levels in the human body in comparison to DNA or RNA, which can be difficult to target using complex gene therapy approaches. Metabolites are fairly simple in that way, in which they can just be added or removed from the human body by modulating either supplementation methods like diet, for example. And so our lab studies metabolites or metabolism as a result of that. And how do we do this? How do we profile the thousands of small molecule metabolites in our blood? We have a long-standing collaboration with Sapient Bioanalytics, a local biotech company that was founded from a lab at UCSD. And the Metabolomics technology uses, as you can see, this automated robotic process to analyze a blood sample containing metabolites. And the blood sample is taken up, and first it is separated such that each of these individual small molecule metabolites is separated so they can be individually analyzed by an instrument called a mass spectrometer. The mass spectrometer will take up every single metabolite and then turn it into a gas so that it can be assessed by its physical properties. And this is the data in real time, where a single blood sample is showing us multiple peaks here that correspond to a different metabolite in the blood of a human. And you can immediately appreciate that there are many peaks there. And what's that telling us? That's telling us that that human blood is rich in metabolites, and that each blood sample contains actually thousands of these molecules. And where do they come from? Well, about 1/3 of them come from our own cells, about 1/3 come from our environment, and about 1/3 come from what we eat. If we look at the data that comes out, and again, I'm stressing how many peaks that you observed in that single representation, and they often overlap each other. So we work to develop AI methods that can actually go into that data, separate each peak, extract it, and determine from the size of the curve how abundant that particular molecule is in a healthy patient, for example, compared to an autoimmune patient. And if you're looking at the left side of this, you can see that here's an enlarged representation of those metabolite peaks, and you can see that they overlap each other. And the AI software will go in and extract one peak and determine the area under the curve. And the area under the curve is proportional to its abundance. And so what we're looking at here is a group of metabolites, some of which that are high and some of which that are relatively lower in the human blood. And what you're seeing in the middle panel is basically just that, is that we can detect metabolites that are very rare all the way to very abundant metabolites in a single blood sample. And not only that, we can identify quite a large superclass of different metabolites that can correspond to lipids or fat-like molecules, to DNA or RNA small molecules, to amino acids or other organic compounds. So we can really capture kind of a diverse representation of metabolism using this method. So how have my lab used metabolomics to understand autoimmune diseases in particular? Well, studying autoimmune diseases in humans is actually quite difficult because they're often considered chronic conditions that flare up in ways that are not entirely predictable. So it can be difficult for a clinician to obtain a blood sample that corresponds to the disease flare-up if the clinician doesn't know when the patient will get sick. So often those samples are collected after the disease has been treated. Because of course, the priority when someone is undergoing an autoimmune disease flare-up is to treat them, not necessarily to get that blood sample. So my lab used a different strategy to study autoimmune disease. You may not know that autoimmune diseases often flare up in patients who receive cancer immunotherapy drugs to treat their solid tumor. A bit of background. In contrast to traditional cancer therapies, such as radiation therapy or drug therapy, which target cancer cells basically just by poisoning all cells in the environment, cancer immunotherapies are immune-based therapies that unleash the immune system to attack cancer cells in a very specific manner. These are drugs such as Yervoy, Keytruda, and Opdivo that have truly revolutionized cancer therapy for solid tumor. In fact, the discovery of cancer immunotherapy received a Nobel Prize in Chemistry in 2018. And so since that time, a number of different cancer immunotherapy products or therapeutics, including a class of antibodies called immune checkpoint inhibitors, have been developed and approved by the FDA for more than 20 different cancer types. Really revolutionary and the gold standard in immunotherapy. But what's the dark side? Well, this activation of the immune system that's achieved by cancer immunotherapy drugs such as Yervoy, Keytruda, and Opdivo also can cause autoimmune complications. And these are inflammatory disorders. And if you look, they can affect almost every different organ system. Some of these are bona fide autoimmune conditions that flare up after taking these drugs. And this really represents the two sides to the immune system coin. On one hand, the immune system can protect us against viruses, bacteria, or cancers. But on the other hand, if the immune response is too strong, it can cause unwanted side effects in the form of autoimmune diseases that flare up right after the cancer immunotherapy is administered to the patients. Perhaps, as we discussed, these conditions can affect any different organ system, but they are predictable because they always arise within weeks after the cancer immunotherapy is produced. And indeed, the cancer patients who go in for their immunotherapy give a blood sample every time they go in for their weekly treatment. And in this way, we were able to design a metabolomics experiment that analyzed the blood and blood metabolites of patients before they received cancer immunotherapy and then out to 12 weeks and after, with blood sampling every 3 weeks. And many of these patients had an autoimmune disease flare up during this time. So we were able to sample metabolites and metabolite changes during this critical period where these autoimmune diseases were flaring up, and even before and even after. So we performed a study of about 150 different cancer patients across 3 different patient groups. And you can see the patient groups are colored in red, green, and blue. And they correspond to patients, for example, with melanoma who were treated with Yervoy, patients with lung cancer in green who were treated with Keytruda for lung cancer, and patients in blue who were treated with either Yervoy, Keytruda, or Opdivo cancer immunotherapies for a variety of different solid tumors. And what did we observe when we looked at the metabolites? Well, we uncovered a specific metabolite called lyso-PC or LPC that we observed to be specifically and uniquely reduced in every patient who developed a severe autoimmune disease after cancer immunotherapy. In fact, I have to stress that every single patient, and there were about 30 patients who developed autoimmunity after their cancer immunotherapy, had a specific decrease of this one metabolite, lyso-PC, in their blood at the time where they were developing autoimmune symptoms. What is lyso-PC? Lyso-PC is a lipid or a fat that is quite abundant in human blood. And what you're seeing here is the LPC chemical footprint that we look for, that we were able to match to a database using the mass spectrometry data that we produced in the patients. And being a fat or a fatty acid, LPC can be is represented in the blood as saturated LPC, saturated fat, or unsaturated LPC. And the specific molecule that we picked up that was missing in the blood of the cancer patients with autoimmunity was unsaturated LPC. They were missing unsaturated LPC. And you must know, or you may know, that unsaturated fats like LPC are considered healthy fats. They are associated with foods that are commonly associated with protection against inflammation, like certain types of fish, like avocado oil, nuts, olives, and other healthy fat-containing foods. So to summarize what the studies of human cancer patients showed us was that a specific decrease or loss of the metabolite LPC in the blood of human patients was observed in patients who developed autoimmune diseases. As immunologists, we next asked ourselves if and how LPC can regulate immune cells, specifically innate immune cells. And we found through a series of experiments in our lab that LPC actually regulates, it inhibits the action and the number of inflammatory neutrophils in the blood. And just to remind you that inflammatory neutrophils are an abundant cell type in the blood that are part of the innate immune system's inflammatory response. And in fact, neutrophils are already associated with several autoimmune diseases, including multiple sclerosis, inflammatory bowel disease, rheumatoid arthritis, and also lupus itself. And actually, the measurement of neutrophils in the blood, called the NLR ratio, is what clinicians use to determine in part if a patient is undergoing an autoimmune flare-up, because the number of neutrophils in their blood will elevate significantly. And this is an indication that the patient is undergoing an autoimmune flare. And truly, these cells are at the scene of the crime. In almost every autoimmune target organ, for example, the brain in MS, or the joint, inflamed joint in rheumatoid arthritis, or the skin in lupus, patients who are experiencing a flare will often have an increased accumulation of pro-inflammatory neutrophils in those affected tissues. So to summarize again, what we observed in humans were that lower levels of LPC in the blood were associated with the development of severe autoimmune complications and an elevation in the blood of neutrophil numbers. And then we also performed a series of experiments in mouse models of autoimmune disease, or in this case, of IBD colitis, to show that if we supplement back LPC, this healthy fat, to raise those levels again to physiological levels, that we could suppress the autoimmune manifestations in these mouse models and also suppress the number of neutrophils. And that's the data that I'm going to show you here. And I'm going to walk you through this data. What you're looking at here in these images are images of the distal colon of mice experiencing ulcerative colitis. And here, these pictures of the distal colon, if we focus on the upper left of the mice, these are the control, the healthy colon. I think you can appreciate that there are two levels here in the histological sample, and there's an organization here of those colon samples. In contrast, comparing directly to the bottom panel, that organization is lost. What does that mean? That's damaged tissue. These tissues have been damaged in the mouse that's experiencing ulcerative colitis, so that discrete organization is gone. You may also appreciate there's many more little black or dark purple dots in the ulcerative colitis sample, and that's indicating an infiltration of pro-inflammatory immune cells, of which in this case, neutrophils comprise a significant proportion. That's the untreated sample. When we re-supplemented LPC to these colitis mice, and we're looking at the lower right-hand panel here, I hope you can appreciate that we were able to prevent a large degree of that tissue damage because we're able to maintain the organization that you can see in the distal colon. And we also decrease the number of infiltrating immune cells. And that's just represented here by an overall toxicity score, where here we're comparing in the open red dots, the colitis mice with a very high, relatively high colitis score of 30, because we're adding up all these measures on the histological scale. And when we supplemented that healthy fat LPC, we can suppress that toxicity. Are we completely inhibiting it? No, this is not a broad scale immune suppressant. That's not what we're seeing. This is a natural anti-inflammatory metabolite that's kind of turning down the flame on that autoimmunity. And we actually think this is a good thing, because we think that LPC administration is not broadly immunosuppressive. In this case, with this degree of immune suppression, this organism we believe can still fight off an infection. Are the neutrophils down? Yes, they are. We also observed when we looked at the NLR that the neutrophilia, which is high in the colitis mice, was suppressed. And remember that neutrophil numbers themselves are a clinical measurement of many autoimmune flare-ups. So in conclusion, what we learned from our studies of humans and mice was that low levels of the healthy fat LPC were associated with increased numbers of early innate immune cells in the form of neutrophils, and were associated with autoimmune complications in patients who were receiving immunotherapy. In contrast, higher levels were associated with control of neutrophil numbers in the blood and no toxicity. So what does this ultimately mean? What is the next step here? How can we use this new information and translate it into potentially a better therapeutic for autoimmune disease? Well, if we think about what we talked about the current methods for immune suppression in autoimmune disease, they rely on steroids or immunosuppressive agents like cyclosporin that are really broadly and strongly immune suppressive. And that leads to kind of the two problems of the commonly prescribed immunosuppressants. They're too strong, and they often elicit severe side effects. And they also don't allow for dynamic control of inflammation. It's a more tailored control that can just reduce the inflammation at the time of the flare-up and not result in broad immunosuppression. We think that LPC can fit the description of one such personalized immunotherapy for dynamic control of inflammation. And we're working with another local San Diego biotech called Intratus to develop a form of LPC that can be applied topically through the skin and allow for efficient transfer and increase of LPC levels in patients either before they're taking their cancer immunotherapy drug, because we know in that case that they have a good chance of developing an autoimmune disease, or someone who's experiencing a flare-up. And we're working with this team as a academic biotech collaboration to see if we can formulate a new treatment like this. And so that's where I'll end, and I'll just acknowledge the funding sources for this work, in part funded by the National Cancer Institute, by the SPARC grant at La Jolla Institute, the program in immunology at UCSD, and the Heights, which is a group of philanthropists who have supported this work. I want to just thank my lab who've done all the work, and you all for listening. So thanks so much.
What a wonderful talk, Dr. Sharma, Sonia. Very important. And I have some excellent questions from the audience. But before I even get to some of their questions, I want to just ask you if you think this LPC, adding in LPC, this precision LPC therapy, can be also applied to the very broad range of autoimmune diseases that are out there in the world that have varied causes, like viruses and bacterial infections. So I'd love to hear what you have to say, or what you think about where that's going to lead us in the future.
That's a great question. And we absolutely do. We do believe that this is a broadly applicable therapeutic in the sense that innate immunity is being upstream of the adaptive immune response is broadly applicable to basically all immune responses. And by virtue of that, all immune diseases, because innate immunity is the foundation for which the more specific responses are built. So we absolutely do. And we actually have performed a preliminary study of patients with autoimmune diseases where we have observed that as a collective, if we combine lupus and rheumatoid arthritis with other conditions, that lower levels of LPC are associated with patients who have these conditions. So we do think that this is broadly applicable simply because neutrophils are so broadly applicable to those responses.
Well, that's fantastic. This is something that's so incredibly desperately needed, really. And it's just, it's amazing and so inspiring to see the progress that you're making.
Thanks so much.
Well, let's see. We have a question from Mark. And his question is, as we age, our immune systems weaken. Are there sex-based differences in this immune response, the decrease in our overall immunity between men and women? And in addition to that, Dr. Sharma, you might want to maybe make a few remarks about sort of what you're seeing as sex-based differences in autoimmune disease in general.
Great question. And yeah, so to begin, absolutely. And anybody who's looked at the numbers for autoimmune diseases, those 5 common diseases that I mentioned in the beginning, like, for example, lupus, rheumatoid arthritis, multiple sclerosis, Hashimoto's, Sjogren's syndrome, they are all sex biased in that the incidence and the prevalence of these diseases show a very strong sex bias in that they are more common in women compared to men. And this runs the range of tenfold in the case of lupus, I think, all the way down to about twofold in the case of RA. So we know that there's a sex bias there. What that sex bias is mostly associated with age, not always, but mostly. Most of the risk increases as we age. And so aging plays a really important part in the development of autoimmunity and in the manifestation of the disease. So we know there's a sex component and we know there's an age component. That's there. We really have to do more work to understand why. I mean, with respect to the sex hormones, estrogen and testosterone, there has been a fair amount of work showing that estrogen, for example, is immunostimulatory and that testosterone in general is immune suppressive. So we know that sex hormones play a big role. We also know that the sex chromosomes play a different role, as Erica mentioned, the X, Y, and disease. For example, the X chromosome contains dozens of immune genes that are involved in regulating cells like neutrophils and others in B and T cells. And women have 2 copies of that. So they automatically are coming with more armaments in that fight. So they're going to have a stronger immune system. And we see that. Whereas, but that's going to make them more susceptible to autoimmunity. But to talk about the age factor, yes, our immune system is educated as we age. And it becomes, the idea is that during kind of the years that we would be most susceptible to viral infections, early adulthood into adulthood, our immune system is constantly being educated to be better. And then it passes a point as we age where it kind of gets overwhelmed. Studies are showing that some of the viruses that are chronic in our bodies. For example, human cytomegalovirus is one of those, one of the main culprits. People who are infected with human cytomegalovirus are being exposed to it constantly throughout their life. And it's educating our immune system, but maybe too much. And our immune system starts to weaken because it's kind of focused only on one or two pathogens that are always with us, as opposed to being pliable to new infections. And so that's an example of the immune system aging, and is something that would be benefit from us to be able to turn down that response, for example, against CMV as we age.
Oh, that's great. What a great explanation. Thank you. And thank you, Mark, for asking that question. There's a question from Giovanna, and you've actually partly, if not mostly, addressed this, but perhaps you'll have other details you'd like to share. Her question is, how do you consider hormonal impact and our epigenetic factors in inflammation first and then diseases?
Yeah, I mean, you know, that we've only yet scratched the surface about the role of sex hormones in regulating, you know, post-genetic factors in human disease. And we talked about it just about, you know, estrogen in general being immune stimulatory and testosterone in general being immunosuppressive. And I think that we have the observation that these hormones are important, that they have effects. We even have the observation that modulating them has effects. For example, younger women who may be on birth control will have higher levels of estrogen or progesterone and lower levels of testosterone, and they seem to have a kind of enhanced immune response. So we've had that observation, but we have to finally make those molecular mechanistic links between what estrogen is doing and how that's affecting our genetic responses. And there are certainly examples of estrogen, for example, regulating certain immune transcription, immune proteins that regulate genes. But I think that's where the field is now, where we really, and that's part of the work that we want to do here at LJI, is finally uncover those mechanisms between what the sex hormones are doing and what's specifically happening in the cell.
Oh, that's great. Thank you so much. And Giovanna, thank you so much for that great question. I want to back up just for a second. And could you tell us, please, the common name for how we would know the human cytomegalovirus?
It's HCMV. HCMV.
And what illness is that we would recognize?
That's
Sort
a
of
very good
fully?
question. And actually, HCMV doesn't cause a lot of disease in humans. In a normal, healthy human, HCMV won't, in fact, cause any disease. It's associated with disease in immunocompromised individuals or in pregnant women. And actually, you know, LJI is actually involved in a huge push called the Human Shield to develop a new vaccine against HCMV, because even though it's not been historically associated with a lot of disease, it is important in causing some chronic problems in our immune system as we age.
That's so interesting. Sylvan asks us, why is rheumatoid arthritis much higher in women?
So great question again. So rheumatoid arthritis is one of those that's actually it's not among the most skewed. It's about a twofold. So if we look at prevalence, about two times as many women as men are affected with RA. So it's actually one of the less sex biased ones, but it is there. And that really goes back to what we're observing in the general innate and adaptive immune system of women is in general more robust compared to an age-matched male control. And for example, we saw this during COVID, right? Like severe COVID disease in the first wave and even subsequent was much more severely affecting males than in females in terms of mortality and morbidity. And in general, women have a stronger immune system. Again, they have the X chromosome, which gives them 2 copies instead of 1 copy of specific immune genes. They have higher levels of estrogen, which is an immune stimulation. So this likely leads to more inflammation in the specific affected tissues like the joint. Although that might be balanced by men who tend to have, because they have a higher muscle mass, might have more wear and tear on their joints. So that might be what's evening that up a little bit, because as I said, the skew is 2-to-1. Whereas if you look at lupus, for example, I think it's 8-to-1.
What a great answer. Thank you so much. And thank you, Sylvan, for that question. We have a question from Jesse. How does your work that you have described so well apply to IBS treatment?
Oh, that's a great question. So there are a few treatments for IBS, and there's a couple in phase 3 and for FDA approval. We think that this treatment is a very good candidate for flare-ups for IBS, in that IBS is a neutrophilic disease, and we think that by LPC supplements can be used to treat IBD by decreasing that neutrophilia. And that's the first place that we want to look at in terms of cancer immunotherapy induced colitis, and then later for general.
Well, thank you. And Catherine asks, why is the LPC application in progress right now that you have mentioned, dermal application versus oral?
Yeah, oh, that's a really good question. I was hoping I'd get that one. We've tried different ways to administer it, and by injection, by oral, as well as by the new one is dermal. And what happens is that the things that we eat, they can definitely have a bearing on the metabolite levels in our blood, but the time that it takes for those metabolites to enter our bloodstream after we eat is often delayed, and during that time they can be modified in our tract, in our gut, for example. And those modifications, they can change how effective it is. So we think that while dietary LPC obviously is really important, and eating healthy fats is of huge importance, we think that during a flare-up, the patient might need a little more of a hit. And this transdermal method is to minimize kind of the time, and also minimize the potential for those metabolites to be metabolized and to be changed.
Oh, that's great. There are so many questions to ask you. It's hard to choose, really. How about telling us a little bit about what factors go into, what are you seeing, and what do you think you are also going to see in the future as being key factors in why some people can grow through their entire lives without any symptoms, and other people get seriously ill in the short run, and then perhaps they then can recover, and others are have chronic disease, subject to more and more serious flare-ups. What causes these kinds of variations in people's responses? I know that you're working on trying to discover that, really. And are there signals that we know already, or that you're starting to discover, that will indicate and show us what a particular individual person is likely to experience?
That's a really good question. And if you remember the slide that I showed you about the inputs of autoimmunity, it really talked about those 3 things. It was genetics are important, environment is important, and the immune system is important. So that already underscores how complex this disease, because we know, for example, that human genetics, we all have a different pattern of genes and gene expression, and then epigenetics or environmental triggers layers on another aspect of complexity. And the immune system itself is what is probably the most complex organ in our body. And that's really why those overlap of those 3 already in each its own complex inputs into autoimmunity are what make it difficult sometimes to predict. But that doesn't mean we can't do a better job. And I think the way to do that is through epidemiological, observational, and profiling studies of the type that we do here at LJI, where we can profile folks who are prone or already we know they have autoimmune diseases, and profile them by metabolites, by immune cells, by the things that we study here to really understand the immune system in order to get biomarkers. Biomarkers tell us a lot. It's a blood test, for example, that can tell us a lot whether someone is experiencing or about to experience a disease. For example, when a person's experiencing a heart attack, they have a blood test for something called troponin, and that's a protein that's released in their blood. So we need that for autoimmunity, and it's more than time. And I think that will go a long way. And what folks can do is, your rheumatologist can be your best friend, and rheumatologists are some of the most, I think, basic research oriented clinicians that I've ever met. They're usually really very passionate about basic research and its translation into the rheumatology practice. So I think that that's a great resource to kind of bridge those two worlds.
Okay. Oh, that's fantastic. And very exciting, really. I mean, thank goodness, and so grateful for rheumatologists. David from the audience asks, are there trials now for LPC that are open to join clinical trials?
So we're working on it. We are at LJI here. We're partnering with some clinical collaborators. We're hoping that there will be soon. We're at the stage right now where we're formulating the delivery, and we really, really, really want to get a phase 1 as soon as we can. So please keep in touch with us with LJI. Please stay tuned, and we will get that information out as soon as it's developing. We're really working on it.
That's fantastic, David. That's so important. I'm so glad you raised that question. Well, we are so excited about your talk, Dr. Sharma. Thank you so much. It was wonderful. We look forward to more. And I want to just make a couple of remarks to everyone to emphasize how important it is to participate in philanthropy and support LJI, which is really this incredible cutting edge, the highest level really of research and brilliance you can find. And we need to support LJI clearly, because that's the way we are going to save lives. That's the way we're going to be able to detect disease, prevent disease, treat disease, and ultimately to be able to prevent disease. That's what they're working on. And it really is in the realm of possibility. Scientists need our support, and we need to participate in supporting them. We need to be active participants in our healthcare. If we are not proactive, then we are subject to forces beyond our control. And especially in this day and age, we really need to pay attention. We need to support science as directly and as frequently as we can. And LJI is just a premier institution. We have so much to be thankful for, all of their very hard work. You've joined, to our audience, you have joined a very special group here today. It's a rarefied group of scientists in the best of the best. They do the most impactful research that there is in the world. I and the Global Autoimmune Institute, GAI, are so extremely grateful to have the opportunity to support LJI and to participate in this panel discussion. Thank you very much. Now, as a reminder, a recording of this webinar will be sent out to you later. And we, looking at this slide, you'll see their contact information. Sign up for regular notifications, and including looking out for clinical trials that might be available to you in the future. Thank you so much, Dr. Sharma. Thank you. What a fantastic presentation. Thank you, audience. Great questions.